Since their discovery in 1928, antibiotics have become a common way of treating infections caused by bacteria, fungi, and other microbes. To treat an infection, a doctor usually prescribes a course of antibiotics, such as penicillin, for us to take. This drug is usually contained in a capsule then swallowed, where it dissolves in the stomach. There, the antibiotic goes to work, destroying all bacteria in the stomach, including the good bacteria, which help our body function. The antibiotic is simultaneously absorbed into the bloodstream, where it is circulated around the body to target any infection present in other body sites, such as the throat or ears. Whether you have an ear or urinary tract infection, the gut is the first point of treatment for bacterial infections. Talk Tech to meet in Europe's leading and most fun tech events, and let us go. While antibiotics are helpful to us, a tendency to overprescribe them is one of the reasons the world faces a crisis of treatment-resistant bacteria. Antibiotic resistance already causes about deaths a year and could cause 10 million deaths by 2050. Antibiotic resistance happens when bacteria acquire one or several genes that allow them to either destroy or avoid an antibiotic's effects. They do this either by developing mutations in a gene or acquiring this gene from another microbe. Resistance then spreads through the body in one of these two ways. Antibiotics destroy all bacteria, including the ones that protect us from illness and disease. So any bacteria with a genetic mutation or a gene that confers resistance to the antibiotic will be able to reproduce out of control until the main bacterial species are present. MIT algorithm discovers antibiotic that can fight drug-resistant diseases, or bacteria can swap their resistance genes with other species of bacteria. In this case, the resistance genes could be given to a new species of bacteria, including the ones that cause disease. This would make treating the infection difficult. Resistance can also be spread from person to person. For example, a person that has antibiotic-resistant bacteria in their mouth could spread these bacteria to another person simply by coughing or by kissing them. Until now, scientists have focused research on the gut, seeing it as the main way that antibiotic-resistant bacteria form and spread throughout our bodies. But our latest study shows that we might be tackling antibiotic-resistant bacteria the wrong way. We discovered for the first time that different sites in the body carry different numbers and species of antibiotic-resistant bacteria. We did this by comparing bacteria in the mouth, which is the first point of entry for antibiotics, with those in the gut, where the antibiotics are released. This discovery will be key for the future of treatment and prevention. Spotting resistance started by looking at the microbiome in the gut and mouth. The microbiome is crucial in understanding antibiotic resistance, as it is the sum total of all microbes, including bacteria, fungi, and viruses, found in a particular part of the body. But only some of the bacteria in each microbiome may carry antibiotic-resistant genes. We compared DNA sequences from different mouths and stool microbiomes collected from people living in China, Fiji, France, Germany, the Philippines, and the US. This allowed us to create an overview of all genetic material in these samples, which we then compared with a database of thousands of genes known to cause antibiotic resistance. An algorithm then helped us reconstruct the genes and remove DNA sequences not responsible for antibiotic resistance. We compared these remaining out fast. You can read full post on tryptophans. News link available in text below.